Hello everyone, welcome to my poster. My name is Siyuan Luo and I'm from ETH Zurich. Today I'll walk you through our work titled On Metrics for Subpopulation Detection in Single-Cell and Spatial Omics Data. So in the field of single-cell omics, computational methods are often benchmarked using multiple evaluation metrics. However, the choice of metrics isn't always made systematically. In fact, many analyses default to a few popular metrics like the adjusted rand index without much justification. So we asked, are these really the best metrics for the job? How can we evaluate methods more effectively? And when different metrics give conflicting rankings, how should we interpret and resolve those differences? So in our study, we consider the evaluation of cell population detection and at multiple levels as shown here, from partition or clusters to cell embeddings, graphs, and spatial domains. These representations form the basis for many downstream analysis tasks, and understanding them helps us uncover general principles for evaluation. Our core evaluation setup is the comparison between the predicted structures and the reference annotations. To assess evaluation metrics themselves, we define a set of desirable properties grounded in biological intuition. For example, homogeneity means a metric should reward correct grouping of biologically similar cells, while completeness means that a metric should penalize splitting similar cells into separate clusters. And in the spatial context, we propose properties like edge tolerance and domain continuity, which I'll explain later. These properties help us assess existing metrics and design new ones. So let's look at a few illustrative examples. Here on the left panel, I'm showing you some examples at the partition level. So we design some toy examples where C is the ground truth and P1 and P2 are two different predictions. In the first row, P1 mixes yellow and the purple classes, while P2 keep them separate. So we say that any matrix that gives P2 a better score than P1 is capturing the property of cluster homogeneity. You can see that here, for most of the metrics, they can reflect this property, except for this WC, which stands for valence completeness. And then in the second row, P1 incorrectly splits the blue class into two clusters, while P2 doesn't. So here, metrics that favor P2 capture class completeness. As you can see from these results, the adjusted rand index capture both properties, while the mutual information, which is denoted by MI, only reflect the homogeneity but not completeness. Then on the right panel, we explore spatial domain detection. In these toy examples, spots are colored by their true class inside each circle and the predicted class at the outline. P1, P2, and P3 all make the same number of misassignments, but with different spatial patterns. Here, an internal metric like Charles, Elsa, and pass, they are sensitive to local structure and can detect differences in spatial continuity, though in different ways. Here, I'm showing you some external metrics like adjusted rand index. For the non-spatial one, which is the original version of ARI, it treats all three predictions as equal since they only look at cluster labels, not spatial layout. We argue that in spatial context, metric should penalize errors but also tolerate some small mistakes near the domain boundaries, like here, because the annotations near the boundaries may be uncertain or there are some transitional states existing. So to incorporate this intuition, we introduce neighborhood smoothed metrics, which smooths the class labels based on the surrounding supports. When applied to partition-based metrics, this approach allows metrics to assign higher score to prediction like P1, where where the error is majorly made at the boundary regions, and it treats P2 and P3 equally because the number of edge errors they have are the same. So here, due to space and time constraints, we cannot cover every result here, but our preprint includes a very comprehensive discussion across multiple data types and metric categories. So feel free to read the full manuscript. And just to highlight a few takeaways from our discussion, we advocate for property-based metrics, especially those operate at finer resolutions like class level or spot level metrics. These metrics offer more transparency and interpretability. We also recommend incorporating graph-based evaluations, which rely less on global assumptions about the dataset and are particularly useful for single-cell high-dimensional data. We developed a bioconductor package called POEM to implement all the metrics we proposed and discussed. Please feel free to try it out. Lastly, to summarize, evaluation is not one-size-fits-all. By using property-based, resolution-aware metrics, we can more meaningfully assess computational tasks in single-cell and spatial omics. If you are interested in learning more, please check out our manuscript or feel free to reach out with any questions. Thank you for listening.